Hey guys, welcome back. So we're going to continue talking about carbohydrates and monosaccharides. But in this video, we're going to focus on monosaccharide reactions, and we're going to see some similarities and parallels to reactivity of alcohols and carbonyls. So for our learning goals for this video, we want to make sure we learn how to draw open and closed forms of monosaccharides. So you've seen that a little bit before. We want to learn how to identify what's known as the anomeric carbon. And this is also where we find our hidden carbonyl. And then finally, what we want to do is predict the products of reduction and oxidation of monosaccharides. So I just want to start with some review. So recall that an aldehyde or ketone can react with an alcohol to make what's called a hemiacetal. So here we have our carbonyl carbon. I'm going to highlight in purple. And then it'll react with an alcohol. So here we just have methanol as our alcohol. And when that happens, we're going to attach our methoxy group and then make a new OH or hydroxyl group. And this new OH is the same carbon and oxygen that were involved in the carbonyl. So this is not an oxidation reaction. So if we look at our aldehyde here, what you can see is that we have two CO bonds. So two carbon oxygen bonds. In this case, it's in the form of a double bond. We still have those two bonds. And then in our hemiacetal, we have that same carbon present, and there's still two carbon oxygen bonds. So this hemiacetal carbon is called our hidden carbonyl. And then in the case of an aldehyde, it can react with a second equivalent of our alcohol, in this case methanol, and then we'll make what's called a full acetal. So carbohydrates contain both a carbonyl and they contain alcohol groups because they're those polyhydroxy aldehydes and ketones. So they can undergo what's called an intramolecular cyclicization to form a cyclic hemiacetal. So here, this intramolecular means that there's only one molecule involved. So essentially, the sugar is going to react with itself. And then the cyclization is telling us that it makes a ring. And the ring will always have either 5 or 6 atoms in the ring. That's just kind of the magic number for making stable rings. Okay. And then that's going to be called a cyclic hemiacetal. Okay. So let's take a look at what this looks like. And in particular, let's take a look at a simplified form. So we're going to get rid of a lot of the extra OH groups just for this example, because it will make it a little bit simpler to see. Okay, so over here on the left side of your slide, we have D-glucose. So we can number our carbons where we have C1, C2, C3, C4, C5, and then C6 down at the bottom, because remember we have that aldohexose. We have our carbonyl up on C1, and then we do have multiple hydroxyl groups going down. So we'll just kind of keep that in mind. Okay, so the first thing we want to do when we're kind of visualizing the cyclicization is just take our D-glucose and then I'm going to redraw it so we can see how the ring is going to form. Okay, so over on kind of our second pane, we haven't done any chemistry yet. We're just drawing a new perspective. So we have our carbonyl carbon. So we have C1, and then we keep going down and around for numbering. C2, C3, C4, and now here we are at C5. So in this image, C6 is not shown. Okay, so just kind of keep that in mind, like we're kind of cutting off part of the molecule just so we can see the ring formation. So I'm going to go over here. I'm going to highlight C5 in orange. And I can do the same on the almost ring structure. And then we also have our hydroxyl group on C5. And that's the hydroxyl group that we're showing here in our second panel. So we haven't quite made the ring yet. Okay, so we're going to make a 6-membered ring. So what that means is there's going to be 6 atoms total in the ring. And so if we count just our ring members, C1 is going to be 1, C2 is ring member 2, C3 is ring member 3, C4 is ring member 4, C5 is going to be ring member 5, and then the 6th member of our ring is going to be the hydroxyl group oxygen that's attached to C5. Okay, so when we make our ring, we have our C1 and that hydroxyl group. So if you imagine this ring is being kind of like flat, so like we're kind of looking at it like a deck of cards where there's like 1, 2, 3, 4, 5, 6, or 6 members of that ring. When we make the ring, we're taking our trigonal planar carbon and we're going to make it a tetrahedral carbon. So remember that there is a hydrogen here, and then we don't have it shown, but that hydrogen is still present in the final structure, the final ring. Okay. So when we kind of break the carbon-oxygen double bond and we make our tetrahedral carbon, that hydroxyl group can either go like up, out, so kind of like up out of the plane of the card, or down. So that's what we're seeing here, is we're seeing 2 different isomers that can form. So let's go through and let's just number the carbons first. Okay. So we have C1. I should use blue for this. So we have C1 and C1. So I'll do the top one all the way around first and then come to the bottom one, the bottom isomer. So you have C1, and then we want to kind of keep going around the same way that we did before. So bending down and going around kind of counterclockwise. So you have C1, C2, C3, C4. There we have a C5. And then we have the oxygen that was part of the hydroxyl group on C5. And then for our bottom isomer, we have C1, which we already labeled, C2, C3, C4, C5, and then the oxygen that was part of our hydroxyl group. So in both cases here, we're making a new cyclic hemiacetal. So these are cyclic hemiacetals. And we can make 2 separate isomers. Okay. And then we still have our carbonyl carbon, our hidden carbonyl. And in a sugar, it's specifically called an anomeric carbon. So that's our hidden carbonyl in our cyclic hemiacetal. So I'm going to go over it in yellow, but just keep in mind there's a lot of colors here now. So we have our anomeric carbon, and that's the carbon that has 2 carbon-oxygen bonds. Okay. So for D-glucose, there are several ring forms that can form. So we can make 6-membered rings and then also 5-membered rings. The perspective on the next slide has changed a little bit. So because in the top example and in the previous example, we're making 6-membered rings, I'm going to draw those as chair conformations. The textbook really likes what are called Haworth projections. We'll see those a little bit in the lecture video. They are also used in a lot of biology textbooks, so you will see Haworth projections probably in the future. Because they are just kind of like an entirely new stereochemical representation, I'm not going to get too much into what the Haworth projection means. It is useful, but I'm going to stick with largely chair conformations for the 6-membered rings. Okay. So from D-glucose, we can make 4 different ring structures. So I hope that one of the trends that you're seeing at this point is that sugars have a lot of structural complexity. So earlier we saw those different hexoses. They're all isomers of each other. That was just 4 examples with our D-glucose, D-galactose, D-mannose, and D-fructose. So those were just a few examples. And now we're seeing more structural forms and isomers that can form. So for D-glucose, there are 2 different 6-membered rings that can form. So we have one where the sugar, the hydroxyl group is going down, and then one where the hydroxyl group is up. I know like here it looks more flat, but it is actually kind of like going up. So we have like one that's very clearly down and then one that is more slightly up in this case, and we still call it up. Okay. So this is our carbonyl or anomeric carbon. The 6-membered rings are what are called pyranose forms. And then in the case of glucose, we can also make 5-membered rings. So for the 6-membered rings, we're going from C1 to C2 to C3, C4, and then from C5, the hydroxyl group on C5 is being used to make the ring. So here we have our C5. We can also make what's called a furanose form. So Furanose. And that's a 5-membered ring that is going to be formed by making a hemiacetal with the hydroxyl group that was on C4. Okay, so we can take a look here and find our anomeric carbon. So what we want to do is look in the ring. So here we have 1, 2, 3, 4, 5-membered ring. So we're looking for a carbon that has 2 carbon-oxygen bonds. So if we go from the O that's in the ring and then look at the adjacent carbons, here we have a carbon-carbon bond, carbon-hydrogen, carbon-carbon, whereas on the right carbon of the slide, we have carbon-oxygen and then a carbon-oxygen bond. So here is our anomeric carbon, or our C1. So in both cases, this is still carbon 1. And then also with the pyranose forms, those are also carbon 1. And then if we keep going around in a circle on the furanose forms, we have C2, C3, C4, and then our hydroxyl group. So here we have 4 different isomers, or another 4 forms of glucose. We have our furanose forms and the pyranose forms. And in both cases, we can have stereochemistry at the anomeric carbon, where the hydroxyl group is either down or up. Okay. So this hidden carbonyl is called the anomeric carbon. Anomers are a type of stereoisomer, which means that we're going to have some nomenclature associated with them. So when the hydroxyl group at the anomeric carbon is pointing up, that is actually going to be the beta anomer. So this is a pretty big typo. Okay. And then when the hydroxyl group at the anomeric carbon is pointing down, okay, that is the alpha anomer. So let's take a look at the example on the next slide. So this is interconversion between the 2 glucose anomers. That's called mutarotation. So these are those Hayworth projections I was talking about. So here, when the alpha D glucose, when the hydroxyl group is down, that's the alpha form. And then when the hydroxyl group is up, that's the beta form. So if we go back to our kind of like bigger slide, we can identify the alpha and beta anomers of our pyranose forms and our furanose forms. There's a lot of like nomenclature and jargon with carbohydrates, just a lot of new vocabulary that we pull in. So here we have our anomeric carbon. The hydroxyl group is pointing down. So this is going to be the alpha anomer. As we if we move around going clockwise, then in our second pyranose form, we have our anomeric carbon. The hydroxyl group is pointing up. Just a little bit on the chair conformation, but we still call it up. So this is going to be our beta anomer. And then we'll keep moving around clockwise. So in our first furanose form, we have the hydroxyl group pointing straight up. So this is going to be another beta anomer. And then in our final furanose form, the hydroxyl group is pointing down. So that's going to be our, that's going to be another alpha anomer. Okay. So here going between furanose forms, these are anomers. Going between pyranose forms, these are also anomers. And then going from a furanose form to a pyranose form is going to be a constitutional isomer. So we're changing where the bonds are, going from a 5-membered ring to a 6-membered ring. So we always have open and closed forms of glucose. So you may have taken other chemistry classes or nutrition classes or biology classes where you got really used to seeing them in the closed form or in the open form. And really they exist in equilibrium with each other. So we can have interconversion between alpha anomers and beta anomers. This is shown here with just the pyranose forms, so just those 6-membered rings. So between the pyranose forms, if we start with alpha D glucose where the hydroxyl group is pointing down, we can make an acyclic form. This is the same as the linear form, which we saw earlier as a Fisher projection. So with our 6 carbons, just kind of all in a line. We're here just kind of like redrawn it so it's more suggestive of the way it'll form a ring. And so we have an acyclic form where we restore the carbonyl from our anomeric carbon, and then the ring can reclose. The hydroxyl group can go either up or down. And so we can make the beta anomer of our pyranose form. So at equilibrium, there's always a mixture of the different forms of glucose. Here we very selectively just chose the pyranose forms. They can also interconvert between the alpha and beta furanose forms as well. Okay. So monosaccharides can undergo multiple reaction types. We're just going to focus on 3 today. So we already saw hemiacetal formation. This is where we're going to make cyclic forms of sugars. And this is very common with hexoses. Because we can make those 5 or 6 membered rings. Whenever you try to make a smaller ring, so if you're trying to make like a 4-membered ring or a 3-membered ring with a smaller sugar, that actually puts a lot of strain on the bonds and a lot of strain on the atoms. So we don't typically see them. It'll be most common to see 5 or 6-membered rings. So you're not going to see them as often with our trioses, tetroses, or pentoses. We'll primarily see them with hexoses. Okay. And then in our sugars, we have carbonyls. And so those carbonyls are going to react the same way that we saw in our organic chemistry sections. So an aldose carbonyl, which is basically an aldehyde. It is an aldehyde. There's just other stuff there as well. So aldehydes or aldoses can be reduced to primary alcohols. And then we'll also see the oxidation of an aldose and like sort of a ketose from a carbonyl to a carboxyl group. Okay. So we already looked at hemiacetal formation. So then we'll look at reduction and oxidation. Okay. So anytime we're looking at reduction or oxidation, we're going to be looking at the acyclic or open form of the sugar because that's where we have the carbonyl that is reactive. So the carbonyl is reactive. The anomeric carbon can't be oxidized or reduced in the same way. So we want to make sure that we have that open carbonyl. So chemically, we can take a carbonyl and we can reduce it to a primary alcohol using hydrogen gas and palladium. So we've seen this before. In the case of D-glucose, we can reduce chemically to make D-sorbitol. We can also do this enzymatically. So this is something that can happen as part of metabolism. The type of enzyme that does this is called an aldose reductase. So it'll take an aldose and then reduce it to a primary alcohol. And the reactants and products are the same. So we take D-glucose, we can reduce with an enzyme instead of in a lab with hydrogen gas and palladium. We get the same product. So we get a primary alcohol, which is called D-sorbitol. And the general form of this type of product is called an alditol or a sugar alcohol. And so this is an alcohol that was made from a sugar. And here in particular, we have this alditol. So an alditol is made from an aldehyde. We can also take our aldose or our aldehyde that's part of a carbonyl of an aldose and oxidize it to a carboxylic acid. This is called, or the product is called an aldonic acid when we do that. So this is really common. It can be done with copper 2+, which is used as part of Benedict's reagent to oxidize glucose to gluconic acid. And one of the things that's really nice about this reaction is you see a very clear color change. So copper 2+ is a very nice blue color in solution. If you took the 30A lab at De Anza, you might have done the hydrate experiment where you take these blue crystals of copper sulfate, heat lightly, drive the water off, and they make kind of like a lighter blue compound. So what makes it that blue color is the copper 2+ ions that are present in the solid. So when we oxidize glucose to gluconic acid, we're starting with 2 carbon-oxygen bonds, and then we're oxidizing to make now 3 carbon-oxygen bonds. So in that case, the copper is reduced, copper oxide forms, and this is like a brick red color. So if you have glucose, add copper, you can see this red form. So that's just kind of nice because you can see it very clearly. And that's generally used as a test for what are called reducing sugars. So glucose is an example of a reducing sugar because it reduces copper 2+ to copper oxide. So when we learned about carbonyl chemistry, we learned that like aldehydes, you can oxidize to carboxylic acids, but ketones, you can't oxidize any further. So there's no, in order to oxidize, you have to break a carbon-hydrogen bond. And for a ketone, we don't have any more carbon-hydrogen bonds to break. So usually it'll stop there. With carbohydrates, because we have all of these extra hydroxyl groups, it does introduce a new type of reactivity. So ketosis can be oxidized, but not directly. So there's this rearrangement step that has to happen first. And this will happen under basic conditions. So if you have OH- in solution, so if you have any base or hydroxide around, fructose, which is a ketose, will undergo what's called a rearrangement. And it doesn't matter how this rearrangement occurs right now, like we're not going to go into the mechanism. But when atoms move rearrange, what it can mean is that a functional group moves. And in this particular case, the carbonyl that's on C2 of fructose can rearrange or migrate to C1. So where we had our hydroxyl group, that's going to move to C2, and then our carbonyl is going to move to C1. When we do that, we make an aldose. And in particular, the aldose that we make is glucose. So we're going to make D-glucose from fructose. That's something that will definitely happen in solution. So if you just have like a sugar solution with fructose, this will happen. This can also happen biologically. There are enzymes that are designed to rearrange fructose into glucose, and that's how we can metabolize fructose eventually. So this is just an interesting example of, I think, the complexity that carbohydrates bring, where they can be very difficult to predict. They don't like to kind of like stay as one thing. So once we made D-glucose, if we add Benedict's reagent with our copper 2+, we can oxidize our glucose to gluconic acid and reduce our copper 2+ to copper oxide. So if you're doing any tests for oxidizing sugars, or sorry, any tests for reducing sugars, so the sugars that take copper 2 plus and reduce it to copper oxide, in that case, you may get a positive reaction for fructose, but it's through this mechanism where it rearranges first into an aldose, and then can be oxidized to an aldonic acid. Okay, so just in summary, carbohydrates will undergo intermolecular cyclicization to form hemiacetals. Aldoses can be reduced to primary alcohols. This will happen chemically or enzymatically. And then aldoses can also be oxidized to aldonic acids with copper 2 plus. Ketoses will also be oxidized, but they have to rearrange first to an aldose, which can only happen under certain conditions.